Hello everyone. So today our topic of discussion is the pituitary gland. What is the pituitary gland? The pituitary gland, also known as the hypophysis, it weighs approximately 0.5 grams in adult life and it measures roughly 10 to 15 millimeter in dimension. And where it is present, we can show in the next slide, here. It produced from the bottom of the hypothalamus at the base of the brain. You can see at the base of the brain, it produced from the hypothalamus and rests in a small bony cavity, the sella turcica in the sphenoid bone, and it's covered by the dural fold called the diaphragma cilia. The pituitary gland is in close proximity to the optic chiasma, the cavernous sinus, and the floor of the third ventricle, and it is heavier in women than in men and becomes larger during pregnancy. So it has two lobes, the anterior lobe and the posterior lobe. Anterior lobe is also known as the adenohypophysis, and the posterior pituitary is also known as the neurohypophysis. So coming on to the next slide, here the adenohypophysis it can be further divided into the pars distalis and the pars tuberalis, where the neurohypophysis is composed of pars nervosa, the pituitary stalk, and the median eminence. The pituitary stalk is also known as the infundibular stalk or the infundibulum, and it connects the posterior pituitary gland and the hypothalamus. So here you can see it's a hypothalamus, here is the optic chiasma, here is the adenohypophysis, the anterior pituitary has pars distalis and the tuberalis, whereas the neurohypophysis has the pituitary stalk and the infundibula, the pituitary stalk, the pars nervosa, and the median eminence. This. Now coming on to the next slide, the hypothalamus has a neuronal connections to the posterior pituitary and the endocrine connections to the anterior pituitary. The pituitary stalk, we just saw it in the last slide, this pituitary stalk. It actually connects the pituitary gland and the hypothalamus, and the hypothalamus is bounded anteriorly by the optic chiasma, laterally by the sulci formed with the temporal lobes, and posteriorly by the mammillary body. Dorsally, it is delineated from the thalamus by the hypothalamic sulcus, and the pituitary stalk descends from its central region, which is termed as the median eminence, and stands out because of its dense vascularity, which is formed by the primary plexus of the hypophyseal portal system. And the long portal veins run along the ventral surface of the pituitary stalk. Now we come to the blood supply of the pituitary. So hypothalamus जो है, it receives its blood supply from the circular filus, whereas the adenohypophysis and the neurohypophysis, they receive their blood supply from the superior hypophyseal artery and the inferior hypophyseal artery. Here. So the capillary plexus of the inferior hypophyseal artery, it drains into the neural sinus although some of these capillaries in the neural stalk form short portal veins that drain into the anterior pituitary. This constitutes only a small fraction of the circulation of the anterior lobe, which is one of the best vascularized mammalian tissue. And the major portion of the circulation arises from the long portal vein. These are formed from the capillary network of the superior hypophyseal arteries that invest the nerve endings of the neurosecretory cells in the median eminence. Here, the portal veins form a secondary papillary network into which the hormones of the anterior pituitary are secreted, and the venous channels from the anterior pituitary they drain into the cavernous sinus, and from there into the superior and the inferior petrosal sinuses and into the jugular vein. Here comes the next diagram. Here you can see that it all drain the venous channels from the pituitary, they drain into the cavernous sinus, and from there into the posterior and into the inferior and posterior petrosal into the jugular vein. Now coming on to our next slide, what is the histology of the hypothalamic neurosecretory cells? So there are three types of the hypothalamic neurosecretory cells: the magnocellular neurons, the cell bodies which are located in the supraoptic or the paraventricular hypothalamic nucleus, they project into the neuronal processes by the pituitary stalk in the posterior neural lobe, and the neurohormones are released from the nerve endings into the blood. These are arginine, vasopressin, and the oxytocin. The hypophysiotrophic neurons, they are thyrotropin releasing hormone, the corticotropin releasing hormone, the somatostatin, the growth hormone releasing hormone, the gonadotropin releasing hormone, and the dopamine. And the projection neurons, they are found in the paraventricular and the arcuate nuclei and in the lateral hypothalamic area. These neurons project to the autonomic preganglionic neurons in the brainstem and the spinal cord. So the microscopic examination, they reveal that the hypophysis has three distinctive type cells. Those are acidophils, basophils, and the chromophils. And this pattern of staining it because of the intracellular hormone-laden granules within the pituitary cells. The neurohypophysis is the extension of the hypothalamus and is composed of the bundles of axons from the hypothalamic neurosecretory neurons intermixed with the glial cells. So here comes the embryology of the pituitary system. So we can see here that the anterior lobe of the pituitary, it develops from the evagination of the ectodermal cells of the oropharynx. So basically, this is the we can say the ectodermal cells of the oropharynx in the primitive gut. It evaginates and it is recognizable at 4 to 5 weeks of gestation. This evagination is also known as the red case pouch. It is eventually pinched off from the oral cavity and becomes separated by the sphenoid sinus. We'll see it in the next diagram. So the lumen of the pouch is reduced to a small cleft, while the upper portion of the pouch surrounds the neural stalk and forms a pars tubularis. This together with the anterior lobe is called the adenohypophysis. And if there are cells which remain, if the cells from the Redcase pouch are left behind and they form tumors, these are called craniopharyngioma. So the next slide shows a better image of the embryology of hypothalamic pituitary system. So here's the oral ectoderm. It evaginates and it is pinched off from the oral cavity. We can see here the sphenoid sinus. We can see here the lumen is reduced to a small cleft, while the upper portion of the pouch, it surrounds the neural stalk and forms a pars tubularis. You can see here. This together in the adenohypophysis and form the adenohypophysis. How the posterior lobe is formed? It develops from the neural crest cells as a downward evagination of the floor of the third ventricle of the brain. So here the floor of the third ventricle of the brain, it evaginates and the lumen of this pouch closes as the site fuses to form the neural stalk, while the upper portion of the pouch forms a recess in the floor of the third ventricle, also known as the median eminence. This. Now, so the acha, we are still left with a thing here that the hypothalamic pituitary axis established by the 20 weeks of gestation. It is very important for the MCQ point of view. Now we go to the physiology of the hypothalamic and the pituitary hormone. So physiology, the functions of the hypothalamus, it include the control of the autonomic nervous system and the regulation of the body temperature, hunger, and thirst, and it has a role in the memory, behavior, and emotional responses. So the anterior pituitary gland, it is regulated by three interacting elements, that is the hypothalamic input, the feedbacks of the circulating hormone, and the paracrine and autocrine secretions of the pituitary itself. So anterior pituitary synthesizes and secretes 6 major hormones. Those are growth hormone, thyroid stimulating hormone, the adrenocorticotropin, follicle stimulating hormone, and the luteinizing hormone, and the last, the prolactin hormone. So the growth hormone is also known as the somatotropins. These are secreted and synthesized and secreted by the somatotropes in the anterior pituitary. These somatotropes they constitute about 40 to 50% of the total cells in the anterior pituitary gland. The growth hormone is a single-chain polypeptide containing 191 amino acids. And it shares structural homologies with the prolactin and the HSG. And we know that HSG is synthesized exclusively in the placenta. So what does the growth hormone do? That it affects predominantly growth, metabolism, cell differentiation, and lactation. Now its effects on the protein metabolism. It increases in amino acid uptake and protein synthesis, and it decreases the oxidation of the protein. Fat metabolism, it stimulates the triglyceride breakdown and oxidation in the adipocytes. And in carbohydrate metabolism, it is one of the hormones that maintains the blood glucose levels within the normal range. So it suppresses the insulin-mediated growth, insulin-mediated glucose uptake in the peripheral tissues and insulin-mediated glucose synthesis that can lead to hyperinsulinemia. Somewhat paradoxically, it stimulates the insulin secretion, and growth hormone administration can lead to hyperinsulinemia. So the growth hormone, it actually exerts its effects directly and indirectly through the family of insulin-like growth factors. So there are three members of insulin-like growth factor, and these are insulin and insulin growth factor 1, insulin-like growth factor 2. So what happens is that they have the metabolic functions, but they also have important roles in the cellular proliferation and the functions of the differentiated tissue. So IGF-1 is actually important for the postnatal growth, whereas the IGF-2 is important in the major fetal growth factor. It plays an important role. Now, insulin plays a role in the fetal growth. A reflection of this role is the development of the fetal macrosome and pregnancies associated with the maternal diabetes. Now, the secretion and the synthesis of the growth hormone is stimulated by the growth hormone-releasing hormone and inhibited by the somatostatin, both of which are produced by the hypothalamus. We can see it in the next diagram. I think we should share it first. Here. So here we can see growth hormone. Growth hormone is released, it goes to liver, there are IGFs and metabolism and the tissues, growth and bone. So what happens is that the somatostatin is an inhibitory hormone. It inhibits the secretion of the growth hormone releasing hormone. So growth hormone will be inhibited. And the somatostatin is actually a 14 amino acid peptide that is synthesized in the hypothalamic neurons, mainly located in the anterior periventricular nuclei. And the factors which affect the growth hormone releasing hormone and the somatostatin include the sleep, stress, exercise, blood glucose level. So going back to our previous slide. Yeah. So the factors affecting these include the sleep, exercise, stress, and blood glucose levels. In addition, the IGF-1 provides a negative feedback to the pituitary glands and the hypothalamus. Finally, the estradiol acts to increase the sensitivity of the tissue to the growth hormone. So we come to the prolactin, and okay, going back to the growth hormone, one important thing to remember is that growth hormone production begins early in the fetal life and it continues throughout the life. Although at a progressively lower rate, main daily growth hormone secretion is pulsatile. It is high in childhood with a peak at puberty and secretion falls during the adulthood. In the adult human, approximately 5 pulses of growth hormone are secreted during a 24-hour period, with a larger peak occurring at the onset of the sleep at night. And half-life of growth hormone in the circulation is about only 20 minutes. Now going on to the prolactin, it is produced and secreted by the electrotrophs in the anterior pituitary and it constitutes about 10 to 15% of the anterior pituitary cells. It is a single chain protein made up of 199 amino acids with the 3 disulfide bridges, whereas when we discussed the growth hormone, it was about 191 amino acids. So estrogen stimulates the proliferation of the pituitary electrotrophs and resulting in an increased quantity of these cells in the especially during pregnancy. So coming on to next slide, it is mainly involved, what does the prolactin do? It is mainly involved in the preparation of the breast for lactation, and it is also notable that some men have same circulating concentrations of the prolactin as a non-lactating women, and it has effects on the hypothalamus, pituitary gonadal axis, and it can inhibit the pulsatile secretion of the gonadotropin-releasing hormone from the hypothalamus, and could alter the activity of certain steroidogenic enzymes. So the prolactin secretion is regulated by a dual hypothalamic inhibitory and stimulatory hormone, and it can also regulate its own secretion through a short feedback loop. So it is pulsatile, prolactin secretion is pulsatile, and it increases with sleep, and damage to the hypothalamic control can cause increased prolactin levels. Dopamine released into the hypophyseal portal veins from the nerve terminals in the hypothalamus, the main neurohormone inhibiting the prolactin secretion. Somatostatin also has inhibitory effects, and stimulatory factors include the thyroid releasing hormone and other releasing factors, pregnancy, lactation, estrogen, opioids, dopamine 2 antagonist, and the sleep and stress. So this is what I told just. Here is a diagram which we have already discussed, that it also, the prolactin has a short feedback loop. It can inhibit itself, and suckling has a positive feedback on this prolactin secretion. Dopamine and somatostatin, they have inhibitory effects, and also the hypoglycemia causes the somatostatin release, and hyperglycemia stops the somatostatin. Now we come to the gonadotrophins, such as the FSH and LH and HSG, HCG, the human chorionic gonadotrophins. They are glycoproteins, approximately weighing 30,000 Dalton, and they are made up of the gonadotrophin-specific alpha subunit and a beta subunit that confers the biological specificity to each hormone. So LH and FSH are secreted from the gonadotrophs in the anterior pituitary, and they constitute about 10 to 15% of the anterior pituitary cell. The receptors are typically G protein linked receptors, and they activate the receptor activation by the gonadotropin binding stimulates the adenylyl cyclase, resulting in the rise in the cyclic AMP levels. So FSH and LH, they are important regulators of the steroidogenesis in the gonads. In the testes, the LH acts on the interstitial or the Leydig cells, whereas the FSH acts on the sertolite cells. So the gonadotropin releasing hormone, there is a signal transduction. LH and FSH are released, they go into the circulation, and the LH acts on the thecal cells and the Leydig cells, and the FSH acts on the granulosa cells and the sertolite cells. As a result, there is oogenesis, spermatogenesis, and the steroidogenesis. Now coming on to the posterior pituitary. So the posterior pituitary gland, the arginine, vasopressin, and oxytocin. They are small peptides with strong structural homologies, and they consist of 9 amino acids that are arranged in a string structure with a short tail. So AVP, the arginine vasopressin, is mainly synthesized in the supraoptic nuclei and to a lesser extent in the paraventricular nuclei. So coming on to the detail of the arginine vasopressin, it's the name actually, it derives its name from the first action described to it nearly 100 years ago, that is increase in the systemic blood pressure. It is a pressor agent. And the actions of the AVP on the water balance in the kidney, where it is effective in very low concentrations, gave rise to an alternative name, the antidiuretic hormone, and the two names tend to be used interchangeably. So coming on to the important diagram I have added from the internet, obviously, this. In the kidney, what happens is that the AVP, it acts on the G protein linked V2 receptors, and on the capillary side is basal side of the distal convoluted and the collecting ducts. It simulates the synthesis of the cyclic AMP. It actually activates the cyclic AMP. It activates a kinase on the luminal side of the ducts, which initiates a series of events culminating in the insertion of the water channels, the aquaporin. You can see here the long-term aquaporin 2 expression is increased, and as a result, water passes into the ducts, into the collecting duct cells and by the osmosis across the basal membrane into the interstitial fluid and hence back into circulation. So there is an osmotic gradient from the cortex to the medulla of the kidney. Thus, as the collecting ducts pass through the cortex to the medulla, the increasing amount of salt-free water can be reabsorbed by the osmotic gradient. So water passes into the circulation. There is an osmotic gradient from the cortex to the medulla. The collecting ducts pass through the cortex to the medulla, increasing amount of the salt solute-free water can be reabsorbed by the osmotic gradient. This is governed by the aquaporins and hence AVP. So increased osmolality of the extracellular fluid stimulates the AVP release. So we can discuss it in the slide. Acha. So it has three receptors concerned with the okay. So yes, this diagram explains it all. So the text in the book actually tells that if there's hypovolemia or the hypotension, that is there's decreased volume in the blood that is just by 5 to 10%, the AVP secretion is stimulated. So this is controlled by so-called stretch receptors that detect the changes in the blood volume. These are the baroreceptors, and for example, if the hemorrhage after there's a hemorrhage, AVP secretion will be stimulated and water will be detained to increase the blood volume. And other stimulants you have, these are the emotional stress, pain, variety of the drugs, and so what happens is there's decreased blood pressure sent by the baroreceptors and there's decreased baroreceptor firing. So it will transfer the signal by the vagus and it will increase activity of the nerves innervating the afferent arterioles. As a result, there will be increased hypothalamic secretion of the ADH, transportation to the pituitary and posterior pituitary and release from the posterior pituitary into the blood circulation. So in the blood vessel, what happens is that it binds to the vasopressin 1 receptors on the smooth muscles of the blood vessels. There will be the ADH will activate the calcium signaling pathway and there will be systemic arterial vasoconstriction. So apart from its primary role in the water conservation, we have just told that there will be vasoconstriction by the calcium and phospholipase C generated second messengers. And the effects of the stimuli, as we know that other stimulants are the emotional stress, pain, and variety of drugs. So nausea and vomiting are extremely potent stimulators of AVP release, and the effects of these stimuli may be important in stimulation situations such as post-operative state, affecting the water balance. By contrast, alcohol is a potent inhibitor of the AVP release, as little as 30 to 90 milliliter of whiskey is sufficient, resulting in inappropriate dehydration and some of the symptoms associated with the hangover. The preventive measure is to drink a steady storative volume of water after excess ethanol consumption. So now coming to here. So we see there's increased osmolality, decreased blood volume. It is a positive effect. AVP is released, and it is balancing the water and the AVP release. Now oxytocin. It is a neuropeptide and it has a wide spectrum of the center and the peripheral effects as a neurohormone, neurotransmitter, or the neuromodulator. It is produced in the magnocellular cells in the hypothalamus and it is also produced in the peripheral tissues like uterus, placenta, amnion, corpus luteum, testes, and heart. The major action of the oxytocin is in lactation through a neuroendocrine reflex that it initiates the letdown of the milk by inducing the contractions of the myoepithelial cells surrounding the alveoli of the mammary gland. And it also has a role in it induces the powerful contractions of the uterine muscle to induce labor. It can also have the stimulatory effects at the AVP-V2 receptor in the kidney, and there's no natural reacting diseases associated with the oxytocin secretion, but in an off situation, when given in high doses as intravenous infusion, 5% dextrose, it can cause water retention and iatrogenic hyponatremia. So, thank you. That's all for today.